my lab. In case you were here Friday, yes, this is the same lionfish shirt. And if you were here Friday, you may be so lucky to gaze upon this piece of art 4 times, because this is only the second installment in the Fantastic Four PhD defenses. So today, Casey Benkwitt is up. Casey got her degree, I hope I said this right, in Bowdoin? No, dang! Bowdoin College in Maine. Prestigious undergraduate institution. She had a lot of experience during her undergraduate and shortly thereafter, including a NOAA REU at Hatfield Marine Science Center, just like Lillian did, except Casey looked at salmon. She also did an REU with Mike Heithouse and his group at Florida International University in Shark Bay, Australia. Perhaps where you got interested in predation. Yeah. And then spent some time teaching at a marine lab in the Florida Keys before starting grad school. She started here in 2010 along with the rest of the fabulous 4, and like the rest of them earned an NSF Graduate Research Fellowship, and then just started earning awards, and there's so many of them, I didn't memorize them all. It just goes on and on, but I'm only going to do the top 4 because it's embarrassing. She earned the Oregon Lottery Scholarship, the Marine Necton Award from the College of Oceanic and Atmospheric Sciences, the Berkeley Marine Conservation Fellowship from the American Fisheries Society, and the UREX Graduate Fellowship here. She's already basically published her dissertation, I think all but 1 chapter, and has 12 publications to date, including in Biological Invasions, Marine Ecology, Progress Series, and Ecology. And that's the boring part of the introduction. I'm going to hand it over to Lillian Tuttle because we need to stretch the time here because we're still waiting for a committee member. So go ahead, Lillian, and take your time.
Go really slow. Okay, so Casey, we love you, so we are going to deliver your roast in Limerick style. There once was a girl from New Jersey who loved the ocean so dearly. She moved to Oregon. Oh, hold on. Oh, there we go. Joined the lab of Hickson. Maybe I'll just use this. Thus beginning her graduate journey. Go Bees! The young scientist's name was Casey, and she was known for being quite brainy. But on tropical isles, she was known for her style, though some might call her fashion sense zany. When Casey sets her eye on a prize, her competitive side starts to rise. From board games on land to lawn games in the sand, with her winning streak, all odds she defies. But all that winning can really make Casey tired. Don't worry, sometimes we're all a bit uninspired. But the next time you sleep next to the dance floor this deep, don't trust friends not. Don't trust friends not to have conspired. Her talents extend past her science, telling jokes night and day that are riots. Though her she thinks they're hilarious. Though her puns might be corny, Though her puns might be corny, after a few dark and stormies, you'll be laughing along in compliance. The future for Casey looks bright, though at this moment she might feel a fright. Casey, after today, it will all be okay. We know you will soar to great heights.
Oh.
Yeah, I saw Lisa look good.
Oh, we're good. Okay, wow. Without further ado, one thing I did want to say, I went on and on Friday about how my lab is mutually supportive, just wonderful support with each other. That's true with Casey unless you want to win a game against her. If you really want to win a game against Casey, just forget it, because so much for mutualism. Okay. Because she always wins. Okay, here we go.
Oh, thanks, Lord.
Aloha. Have fun.
Thanks.
Okay. Hi, and thank you to Mark and my lab mates for that lovely and super funny introduction. And thank you all for coming to my defense today. For thousands of years, humans have been both intentionally and accidentally introducing species to new locations around the globe. However, in recent years, increases in global shipping and transportation have led to an increase in the spread of introduced species that far outpaces anything in history. For example, it's estimated that at any one time, over 7,000 species are traveling around in ballast tanks of ships on the world's oceans. Now, not all of these introduced species become established in their new locations, but this rate is also increasing. For example, it's estimated that over 120 new species will become established in the United States between the years 2000 and 2020. Now, those introduced species that become established, spread, and cause some sort of ecological or economic harm are typically called invasive species. And studying these invasive species is important for several reasons. First, from the perspective of basic science, they offer natural experiments to test questions of general evolutionary and ecological importance that would otherwise be impossible to test at such large scales in the field. From the perspective of management, studying invasive species is important because they can cause harm to ecosystems through disrupting native food webs as well as altering ecosystem function. From a human perspective, invasive species can cause harm to human economies as well as human health. Population density, or the number of individuals per unit area, is important at all stages of the invasion process. It influences the likelihood of their establishment, their rate of spread, as well as their overall effects in their new communities. Therefore, one central theme of my dissertation research is how does population density influence an invasive species in terms of both the invader itself and its interactions with native species. Now, the negative effects of invasive species are caused not just by their high population sizes, but also by their strong negative per capita effects. In particular, invasive predators have stronger effects than native predators and have caused a decline in many native prey species. Therefore, a second goal of my dissertation research was understanding the spatial extent and the mechanisms behind the effects of an invasive predator. Specifically, we'll be journeying underwater and investigating the effects of an invasive predator on and around coral reef ecosystems. Now, coral reef fishes are an ideal study system for many reasons. First, they've long been used as a model, in part because juveniles and adults are easily seen and manipulated. Coral reef fishes are also important ecologically as the most diverse vertebrate assemblage in the world, and economically as a source of food and tourism revenue. Also, from my perspective, studying coral reef fishes was way nicer than diving off the coast of Oregon, because I got to go to the Caribbean, which is pleasant and warm and clear and great. Now, like most ecosystems around the world, coral reefs face a number of threats, including pollution, habitat destruction, overfishing, and climate change. However, there is only one fish that is considered invasive on coral reefs, and that is the red lionfish, Pterois volitans. Red lionfish are originally from the Pacific Ocean, but they're really popular in the aquarium trade, and it's easy to see why. They have these venomous spines on the top and bottom, these really cool frilly pectoral fins on either side, and these kind of funny little goatees around their necks. And it's through either accidental or intentional aquaria releases that lionfish first made it to Florida in the mid-1980s. Their populations really didn't do much until the early 2000s, when they began spreading up the east coast of the United States. In 2004, they made it to the Bahamas, and from there they've been spreading rapidly throughout the Caribbean and the Gulf of Mexico. And accompanying this rapid range expansion have been nearly exponential population growth rates. This video is from one of my patch reefs in the Bahamas, and you can see it's just completely covered in lionfish. And unfortunately, scenes like this are not uncommon in their invaded range, where they reach a maximum population density of about 400 fish per hectare, compared to just 26 fish per hectare in their native Pacific range. And the big concern with lionfish, in addition to their high population densities, are their strong negative effects on coral reef fishes through direct predation. Lionfish are unique predators in the Atlantic. They slowly stalk and herd their prey with their pectoral fins before swallowing it whole. In the Bahamas alone, lionfish can consume over 56 species from 25 families of reef fishes, and as shown by my advisor, Mark Hickson, and my former lab mate, Mark Albans, even a single lionfish can cause an 80 to 90% reduction in native fishes on coral reefs in as few as 5 weeks. That brings me to the specific goals of my dissertation research. I addressed how lionfish density, or the local number in a given area, influences both lionfish themselves in terms of their demographic rates and their behavior, and how lionfish in turn influence native fishes both on coral reefs and in surrounding seagrass habitats. I also looked at 2 different mechanisms of lionfish effects: consumptive effects, meaning directly consuming their prey, and non-consumptive effects through causing changes in prey behavior. So let's start by looking at how lionfish density influences their demographic rates. So here's a little cartoon showing 2 populations of mesopredators, one at low density and one at high density. At higher densities, we might expect more intense competition for food, like these little prey fishes, and also more intense competition for refuge space from top predators. This could lead to higher loss rates, as well as higher emigration rates away from areas with high population densities. Conversely, in areas with lower population densities, we may expect higher immigration rates, as well as higher individual growth rates, again due to lower competition for resources. Graphically, if we look at the per capita or proportional rate as a function of population size, we would expect an increase in loss rates, including mortality and emigration, and a decrease in gain rates, including birth, immigration, and growth, as population size increases. And such direct density dependence is necessary for population regulation. So this means it prevents populations from going extinct and also prevents them from growing indefinitely. But we know invasive species have often escaped natural forms of population control and have reached really, really high densities. So it's important to test for the presence of density dependence and the mechanisms of density dependence in populations of invasive species. So I tested whether there was evidence of density dependence in lionfish loss rates, which is a measure of mortality plus emigration, recruitment of small juveniles, immigration of larger lionfish, and growth of individual lionfish in terms of both length and mass. To do this, I traveled to the Exumas in the Bahamas and specifically was working near Lee Stocking Island, where there's this really nice matrix of artificial and transplanted reefs that were set up by my advisor and his postdoc Mark Carr back in the early 1990s. Now these reefs offer an ideal setup for conducting manipulative field experiments because each reef is identical in size at 1 meter cubed and all equally spaced 200 meters from the nearest other reef. So here's an example of one of the artificial reefs on which I was working. You can see since the time it was built, it's become completely covered in corals, sponges, and algae, and is home to an abundance of native fishes. So I collected juvenile lionfish from the surrounding reefs and transplanted them to these reefs at a range of densities. Here are two of the lionfish that I collected. You can see circled are tags with elastomer, so they're basically like little tattoos. So I could tell the lionfish apart in order to track individual persistence and growth rates, and also tell when new lionfish arrived at the reefs. I also really like this picture because this lionfish over here, its stomach is completely bulging. So even though it was only about that big, it's already living up to its reputation as a really hungry predator. Here is my experimental setup at the beginning of the study. I have lionfish densities ranging from 0 to 12 per reef, and this represents a range of both naturally observed lionfish densities as well as artificially enhanced. And we did this because lionfish populations are continuing to grow, so we wanted to be able to project a little bit into the future. First, we'll look at lionfish loss rates. So up there, the time in weeks will tick away. We see within the first 2 weeks, a few lionfish were lost from the 3 and 5 treatments, and then not much happened until week 6, when one lionfish was lost from the 7 treatment, and one actually made that swim across the 200 meters of open sand and seagrass from the 10 treatment to the reef with one lionfish present. However, overall, these loss rates are really, really low, and they did not depend on lionfish density. So here's the experimental setup again, this time with the average density of lionfish on each reef throughout the course of the experiment, so taking into account those minor changes in lionfish loss. And now we'll look at the recruitment of small juvenile lionfish, so these are less than 5 centimeters total length, to the reefs. And there were between 0 and 3 recruits to each reef, but again, this did not depend on lionfish density. Next, we'll look at the immigration of larger lionfish, so these are ones that would have swam to the reefs from surrounding areas. And again, there were really low immigration rates with only 5 total new lionfish. However, all of these arrived at reefs with 4 or fewer lionfish already present. So although this pattern is not statistically significant, it may still be biologically important that lionfish only immigrated to reefs with fewer lionfish already there. Finally, this plot shows growth rate. On the y-axis, we have growth in length in millimeters per day. And on the x-axis, we have lionfish density on each reef. And unlike every other metric, there is a significant decline in growth rate with increasing lionfish density. And this was true for both length, as shown here, as well as for mass. So visually, these differences in growth rate look something like this, where a lionfish on a reef by itself grew 1.7 millimeters and 1 gram per week faster than a lionfish on a reef with 9 others. So what does this all mean? As lionfish grow, their diet shifts from primarily invertebrate-based to primarily fish-based. So the faster growth rates observed on reefs with fewer lionfish may have implications for which prey species and which prey sizes are vulnerable to lionfish predation. As fish grow, their fecundity or egg production also increases. So reductions in juvenile growth can lead to lower fecundity and can eventually lead to lower population sizes. However, lionfish loss rates were not density dependent, at least over timescales typical of other reef fishes. Therefore, manual removal by divers is likely to remain the most effective management strategy for the foreseeable future. Also, given the higher immigration rates to reefs with fewer lionfish present, we should likely maintain these removals at regular intervals to prevent lionfish immigration from swamping out any effect of removal efforts. So the overall message from my first chapter is that although population gain and loss rates were not affected by lionfish density, they did have decreased growth rates on reefs with more lionfish present. Now this decreased growth is indicative of within-species competition, likely for food, which means that lionfish populations may eventually become self-limiting simply by eating themselves out of house and home. Now within-species competition and density more generally can also affect the behavior of animals. If we go back to our cartoon fish populations, at higher densities, where there's more intense competition for food, predators may have to spend more time actively searching and hunting for prey. Alternatively, or in addition to spending more time hunting, predators may increase their foraging range at higher densities. This could enable them to exploit food resources that have not yet been depleted or to avoid aggressive interactions in those areas with the highest densities. Therefore, I predicted that if there is within-species competition for food among lionfish, then at higher densities, lionfish will spend more time active, including hunting, and also increase their foraging range. So to test these predictions, I traveled to Eleuthera in the Bahamas, and this time I was working in Rock Sound, which is a shallow sand and seagrass bank with coral patch reefs scattered throughout. I selected 16 reefs with a natural range of lionfish densities of between 1 and 16 lionfish per reef. And I visited each of these reefs at 3 times a day, dawn, midday, and dusk. I chose these 3 times because lionfish tend to be most active at dawn and dusk and typically are inactive in the middle of the day. When I arrived at each reef, I first counted all of the lionfish that were present and immediately recorded their behaviors. I then randomly selected 2 lionfish per reef to conduct focal observations on, where I watched the lionfish for 10 minutes straight and recorded all of its behavior. So the behaviors for both of these metrics were resting. When lionfish were inactive, there's one hidden right there and another one under the ledge there. This was super, super boring to watch for 10 minutes because they didn't move at all. The next behavior is hovering, like this lionfish here and that one there, where they're out, oriented parallel to the seafloor, but again, not moving too much. Next was swimming, when lionfish were actively moving from one location to another. And finally, hunting, when lionfish were oriented head down with their pectoral fins flared. And in this case, there's a little bicolored damselfish that keeps popping in and out of the reef right there. It's okay, he didn't get eaten. So also to get a measure of lionfish movement, I recorded any time a lionfish arrived at or departed from each reef. So first, let's look at the proportion of lionfish that were active. This includes those that were hunting, swimming, or hovering as a function of lionfish density on each reef. And I'm only going to show the results from all of the lionfish on each reef because those focal observations had really similar patterns to the results I'll show here. First, at dawn, the majority of lionfish were active regardless of lionfish density. So each point represents one reef, and the dotted line indicates that there is no significant effect of lionfish density at that time of day. At midday, way fewer lionfish were active, but again, this did not depend on lionfish density. But at dusk, a significantly higher proportion of lionfish were active at higher lionfish densities. Now, in contrast, when I looked at lionfish hunting only, so excluding those that were swimming or hovering, there was no significant effect of lionfish density at any time of day. So these results suggest that lionfish at higher densities must spend more time actively searching for prey, which they do primarily at dusk. So these short video clips will show what I mean by lionfish movement. Here we have a patch reef, and you can see lionfish starting to swim away into the open sand and seagrass. Now these reefs were isolated from the nearest other reef by several hundred meters, so they're really just moving into areas without much structure around. You see that one on the end, turns around and decides to head back to the reef, forget all of you, and everyone else keeps going. This video is also taken at dusk, but this time by an automated camera, so there are no divers present on the reef. And again, you can see two lionfish, this time slowly hunting and hovering as they swim away from the reef. And these behaviors were really typical at dusk, whereas at dawn, I often observed the opposite pattern of lionfish arriving at the reefs from surrounding areas. To quantify these movements, here we have the proportional change in lionfish density on each reef as a function of the maximum lionfish density on that reef. Any points above that dotted middle line will indicate lionfish arriving at the reef from surrounding areas, and any points below the line will indicate lionfish departing from the reefs, as we saw in those last two video clips. First, at dawn, we can see that the majority of lionfish were arriving at the reefs, and a significantly higher proportion of lionfish arrived at higher lionfish densities. In the middle of the day, there wasn't much movement, no matter what. And at dusk, we see the opposite pattern as at dawn, where a significantly higher proportion of lionfish were departing from the reefs at higher lionfish densities. So combined with the results of slower growth that I saw in my first experiment, these results indicate that lionfish are likely experiencing within-species competition for food. Now this can be manifested either through aggressive interactions or direct fighting, or indirectly through resource depletion. And these results may also have implications for which prey species are affected by lionfish predation. Specifically, if lionfish are foraging in the surrounding habitats when they move off of the reef, then prey fishes that inhabit these seagrass beds may be susceptible to lionfish predation, especially at higher lionfish densities. So overall, there is increased activity and movement on reefs with more lionfish present, at least during certain times of day. So now we'll move on and look at how lionfish density influences native fishes on coral reefs. In order to scale up the effects of a predator, we typically assume that if one individual has a certain influence on prey populations, if we add another predator, it will have the same effect, and so on and so forth. So graphically, this looks like a linear decline in the abundance of the prey as we increase the abundance of the predator. However, intraspecific interactions can cause non-linearities in this relationship. For example, competition can limit a predator's consumption rates at higher densities, such that its effects on prey level off as we increase predator abundance. Alternatively, cooperation can cause predators to have disproportionately large effects at higher predator densities. And this could occur through strategies like cooperative hunting that can increase the per capita consumption rate of predators. And there's actually several species of lionfishes in the Pacific that have been documented to use cooperative hunting strategies. Now, understanding the shape of this relationship is really important for invasive predators. If managers set a target density of the predator at the point indicated by the arrow, this can have a large influence on prey populations if the curve follows that top orange curve. However, if the relationship follows the bottom curve, then removing the same number of predators will have very little influence on native prey populations. So to test how the abundance of lionfish prey scales with lionfish density, I used the same experiment that I described for my first chapter. As a reminder, I worked at Lee Stocking Island, and I manipulated lionfish densities on small artificial reefs to be 0 to 12 lionfish per reef. And I returned to the reefs once a week and counted the abundance of all native prey fishes that were there. For the rest of the talk, I'm going to just focus on prey fish that are 0 to 5 centimeters total length, because those are small enough to be consumed by a range of lionfish sizes. Here we have the proportional change in number of native fishes over time in weeks. And if we first looked at the reefs without any lionfish present, we can see that there is an increase in native fish abundance over time. And this makes sense because we conducted this study in the summer when there's high recruitment of small juvenile fishes to the reefs. In contrast, on reefs with just one lionfish present, there's a significant reduction in native fishes. Again, what we expected based on work by my advisor and my lab mate, Mark Albans. As we added more lionfish to the reefs, we see additional decline in native fishes through time. But what we really want to know is the relationship of that curve, so we're going to focus in on the final time point. Here again, we have the same y-axis with proportional change in number of native fishes, this time as a function of lionfish density on each reef. And the best fit curve was a nonlinear decline, where there is a big change in abundance between reefs with no lionfish present and reefs with just one lionfish present, and the effects of lionfish leveled off as you added more and more to each reef. Visually, here are what my reefs look like at the beginning of the experiment. I then added lionfish to some of the reefs, and I'm only going to show 4 of the treatments because otherwise I would have gone completely crazy copying and pasting fish into PowerPoint. Because here are what the reefs look like by the end of the experiment. So up here on the top left with no lionfish present, there are about 70 native fishes by the end of the summer. And this number drops progressively until we get down to the reef with 12 lionfish, where there's just one poor little goby left on that reef. So in the case of lionfish on patch reefs, it seems that the first is the worst. There is the largest drop in abundance between reefs with 0 lionfish and reefs with 1 lionfish present. In terms of management, this means that in order to have the largest effect on prey populations, we should likely set a target density as close to 0 as possible. These results are also consistent with the results of my first 2 studies and are indicative of within-species competition for food. So from this chapter, we learned that lionfish have decreased per capita effects on reefs with greater lionfish densities. But we also know that lionfish are exhibiting movements between the patch reefs and surrounding habitats, especially at higher densities. So how do lionfish affect native fishes in these other habitats? Once predators deplete food on the reefs where they are residing, as I mentioned before, if there's food in the surrounding habitats, they may broaden their foraging range. In terms of prey fish populations, this can cause halos of prey depletion, where prey becomes depleted close to the predator's shelter site first, and then they forage at progressively greater distances away from the reef. However, eventually the costs of traveling further from a reef will likely become too great, either in terms of energy or increased predation risk, so eventually this halo of prey depletion stops. Therefore, I predicted that lionfish effects will occur on main reefs first, where the lionfish are actually living, and then in the surrounding habitats. I also predicted that lionfish effects will decrease with increasing distance from patch reefs. I traveled back to Eleuthera, and specifically Rock Sound in the Bahamas, and this time I selected 16 reefs, all of which had lionfish present on them at the beginning of the experiment. I removed lionfish from half of the reefs and maintained these as low lionfish reefs, and maintained the other half of the reefs as high lionfish reefs throughout the course of the study. I returned to each reef once a week and counted prey fishes both on the main patch reefs and in the surrounding habitats. Specifically today, I'll focus on two of these habitats: satellite coral heads, which were small patches of coral rubble or live coral heads that varied in distance from 4 to 25 meters away from the main reefs. Now, these differed from the main reefs in that they were much smaller, with a maximum size of only 5 meters squared compared to 43 meters squared, and they also had no resident predators, lionfish, or native predators. I also conducted transects in the open sand and seagrass, and these extended out to 35 meters away from each reef. Along these transects, I counted fish in 2 meter squared plots located at 5 meter intervals. And these differed from the satellite coral heads in that there really wasn't much structure around on either side of these. So these next few plots will all have the same axes, with change in fish density on the y-axis and time in weeks on the x-axis. And first, looking at the effects of lionfish on main patch reefs. Again, on low lionfish reefs, over time there is an increase in prey fish density throughout the course of the experiment. And on high lionfish reefs, there is a significant reduction in prey fish density. Now, what's important to note from this plot is that the effects of lionfish began in week 3 and then persisted throughout the rest of the experiment. So next, looking at the surrounding habitats, first those satellite coral heads, we see again there's a significant reduction in prey fish density on satellite coral heads around reefs with lionfish present. But in contrast to the main reefs, these effects did not begin until week 6 and then persisted for the remainder of the study. However, in those open sand and seagrass areas, there is no significant effect of lionfish treatment throughout the entire experiment. Now, there are really low prey fish densities in these areas regardless of treatment, so this may partially explain the lack of lionfish effect. Finally, we wanted to see whether lionfish effects decrease with increasing distance from the main patch reefs. So here we have the same y-axis with change in fish density, and now distance from main reef on the x-axis. And we'll look just at those satellite coral heads. Contrary to my prediction, there is no significant interaction between lionfish treatment and distance, indicating that lionfish effects do not level off as we move further away from the patch reefs. Now, these satellite coral heads extended out to 25 meters from the reefs. I also looked at standardized conch shell habitats extending to 31 meters from the reefs and saw the same exact pattern. This is in stark contrast to native predators whose effects level off within about 5 meters from main reefs. So this cartoon shows what a low lionfish reef looked like by the end of the experiment. On the reefs with lionfish present, we saw that they first caused reductions in native fishes on the main reefs where they were residing. and then caused about a 60% reduction in native fishes on the satellite coral heads in surrounding habitats. Based on behavioral observations, the majority of these lionfish then return to the same patch reef after each foraging bout. In terms of management, we already know that lionfish removals can reduce lionfish densities and help maintain prey fish populations on main patch reefs where lionfish live. However, this research shows that lionfish are also affecting prey fishes across multiple habitats away from where they are residing, so removals might have more benefits by maintaining prey fishes in these multiple habitats. So overall, there were fewer native fishes around reefs with lionfish present. Finally, I looked at whether lionfish have any non-consumptive effects on prey behavior. So we'll return to our little fish friends one last time for a brief background on the coral reef fish life cycle. Reef-associated adults spawn eggs that go up into the water column and become larvae that drift around for about a month. Then when it's time to undergo settlement or recruitment back to the reef, there are a number of cues that influence where reef fishes choose to settle. Specifically, several species of coral reef fishes have been shown to recognize and avoid cues from predators during settlement. And it's easy to see why this would be advantageous, as reef fishes suffer about 55% mortality due to predation within the first few days after settlement, and this transition has been called the predation gauntlet. However, many prey species may be naive to the threats posed by novel predators, like these poor penguins if polar bears were to make it to Antarctica. Now, this prey naivety will generally lead to high consumptive effects, but few non-consumptive effects of non-native predators. Therefore, I predicted that there would be lower settlement to reefs with native predators compared to reefs with invasive lionfish if prey fishes are naive to the threat posed by lionfish and therefore avoid the native predators but not lionfish themselves. To conduct this study, I traveled way to the southern Caribbean and worked off the coast of a tiny island called Bonaire, specifically on a sandy flat in the shallows. And on this sand patch, I built 15 reefs made out of coral rubble. Next to each reef, I placed 2 cages in which I could house predators. That way, settling reef fishes were exposed to the cues from the predators, but could not actually be consumed by them. I arranged these reefs into 3 blocks, each with 5 reefs with the following treatments: empty control with no cages, empty cage, invasive lionfish, and then 2 native predators, the French grunt, which primarily eats invertebrates, and the Graysby grouper, which is the most ecologically similar to lionfish and primarily consumes small fishes. So at night, usually around the new moon, there would be a pulse of recruits to these reefs. And then every morning at dawn, a pair of divers went to the reefs and we counted and removed all of the recruits from the night before. We also fed all of the predators. The lionfish and the Graysbys got a diet of damselfish, and the grunts got a diet of shrimp. The day then went on. Divers returned to the reef around dusk, and this time randomized the treatments within each block. And it turns out that little fish loved these rubble patches. There were over 2,500 recruits in 16 days of over 35 species. But today I'm just going to focus on mahogany snapper, which are highlighted in the circle hovering above the reef there. So here we have the proportion of snapper recruits as a function of treatment on the x-axis. And we see that there was similarly high recruitment of snapper to reefs without any cages, the empty cage control reefs, as well as the invasive lionfish reefs. In contrast, there is significantly lower settlement to reefs with both the native predators, the grunt and the greaseby. Now, I repeated this experiment, but fed the lionfish and the greaseby's mahogany snapper instead of damselfish, and I saw the exact same pattern as shown here, where regardless of predator diet, there is about a 52% reduction in settlement to reefs with native predators compared to invasive lionfish. Across both experiments, there were over 400 recruits to reefs with lionfish compared to just over 100 to reefs with greaseby, which proportionally looks something like this. This is a problem because native fishes like mahogany snapper may be getting hit by a double whammy, where they're more likely to recruit to reefs with lionfish present, and once there are more likely to get consumed by lionfish because they have higher consumption rates than native predators. So overall, there is higher settlement to reefs with lionfish, which is indicative of no non-consumptive effects of lionfish on coral reef fish recruitment. Overall, this research clarifies the extent and the mechanisms behind the effects of invasive lionfish. And I already discussed the management implications with each chapter, but in general, this research also reveals that invasive lionfish are really different than native mesopredators. They do not experience density-dependent population gain and loss rates, at least at timescales typical of other reef fishes. Not only do they cause large reductions in prey fishes on coral reefs, but they also have broader foraging ranges than native predators that encompass multiple habitats. And lionfish are unrecognizable to at least some species of coral reef fishes. This also means that typical anti-predator strategies may be ineffective against lionfish, including avoiding reefs with predators at settlement and inhabiting small structures in seagrass beds away from coral patch reefs. Overall, these characteristics may at least partially explain why lionfish have such strong negative effects on native coral reef fishes and are likely shared by at least some other successful invasive species. More broadly, by examining how the ecological effects of invasive predators vary with differences in local population density, habitat, and non-consumptive effects, we can gain a more complete understanding of how predators influence native prey populations. This information is important both from a management perspective due to the continued increase in number and severity of introduced predators, as well as from the perspective of basic science because it broadens our understanding of how predation influences communities across a variety of contexts. I'm going to start to cry already. Okay. So I have a lot of thank yous to do. Yeah. First, this one's not emotional, it's just money. But it's really important. So I was really lucky to have a lot of different funding sources. So thanks. Mark. So happy you finally made it back to Hawaii. And even though we've been living almost 6,000 miles apart for the past few years, you've been a really great advisor. Taught me many things, like don't email embarrassing pictures of yourself to your graduate students. And also, I've been thinking about this one a lot lately. It was one of my first lessons. Is that fish always point in on PowerPoint slides. But really, thank you for being so inspiring and for pushing me and for being awesome. I'd also like to thank the rest of my committee, Lisa Madsen, Selena Happel, Scott Happel, and Bruce Mangy. Thanks for all your encouragement and your support and your insightful comments. Hopefully you'll have a few more, but not too many in the next And thanks to all the other faculty and staff at OSU and IB who've helped throughout my time here, especially the awesome office ladies who've helped me navigate the bureaucracy, which I still don't understand,
And thanks to Kevin, our DSO, for dealing with the thousands of dives that our lab does every single year. So, really lucky to have an awesome group of lab mates. We spend all summer on tiny islands together and then come back to Corvallis and still want to hang out. And I've even lived with several of you, and I still love you guys. So thanks for all your help in the field, especially that one summer when we didn't have any undergrad assistants, and I decided I wanted to look at lionfish behavior. So you all got up at dawn with me and then skipped dinner with me, which is really nice. So you guys are awesome scientists and amazing friends. There's a lot of other people who helped with the field work at many different research stations, so thanks to all the other assistants who were my dive buddies, who hauled around rubble and conch shells with me underwater, and who navigated logistics of boat use and everything else that goes into a field season. And also thanks to, I had a few awesome undergrads who helped me enter data back in Corvallis, which is a lot less glamorous, but really, really important too. So thanks to all of them. Okay, I think I'm okay now. Okay, so thanks to all my friends, both here in Corvallis and down in the Caribbean, some of whom I hope got to watch today. Thanks for all the awesome adventures, and I'm really excited. Hopefully we'll keep, seeing each other in the future. So Pat, thanks for being one of the 6 people trapped on a deserted island with me during my first field season. I'm really glad you're one of them. Thanks for all the dance parties and all of your support and help being my dive buddy and listening to me go in circles about how to write one sentence and everything else over the past 5 years. So you're probably as happy as anyone that I can stop being such a stress case now, hopefully. And then finally, thanks to my family, my mom, my dad, my sister Allison, and her husband Matt, who flew all the way out here to see this today. Obviously, you guys have been supporting me my whole life, and I wouldn't be here without you. So, thank you. And I'm really happy that now when we talk on the phone, you don't have to ask me what I'm doing, and I don't have to explain why it's taking 6 years. And then I think we have time for a few questions if anyone has any.
Questions for Casey?
Yeah, Cami.
So I was curious about the juvenile recruitment. If you could just talk a little bit more about that. I think you were saying that they were showing up at reefs where there was high density as well, or there wasn't a significant threat there.
For the recruitment of lionfish or the recruitment of other fish?
Sorry, the recruitment of lionfish. If you could just talk a little bit about their life cycle and maybe what we could learn from that as far as benefits.
Yeah, so lionfish have a really similar life cycle to other reef fishes, so they also have that pelagic egg and larval stage, although their eggs are in this crazy mass that seems to be pretty protected from predation. But aside from that, we don't know much about especially those early settlement stages in either their native range or the invaded range. We have a hard time finding the recruits. So the ones I saw were all about 2 centimeters. But yeah, there was no difference in recruitment to reefs with different lionfish densities. So it seems that at least in terms of that metric, there's no density dependence, so it doesn't really make a difference. But if anyone can find lots of recruits, let us know, and our lab will gladly study them. So. Yeah, Neil.
So I'm wondering how effective the lionfish removal by scuba divers is, given these things seem like the perfect predator for where they are, given that the recruits don't recognize them and they have bigger distance effects than the natives. It seems like it's a ticking time bomb if the time bomb already hasn't exploded.
Yeah, you're absolutely right. So we are definitely not going to be able to get rid of lionfish in the Atlantic. But what we have seen is that at least over limited scales, the removals can help maintain certain areas of native fish populations. So targeted removals in really important habitats, or maybe marine protected areas or something like that, can be effective. But lionfish are crazy. In addition to their strong and high predation rates, they live almost anywhere. So they've been found in estuaries and down to over 1,000 meters deep. So there's no way that manual removals by divers is going to, stop the invasion completely. All we can hope is to have benefits in certain areas on certain prey fishes. Yeah, Chincha.
There was some of your work that's taking place on catch reefs. Do you think the effect would be greater on reefs that are more expensive?
Yeah, so I think that probably the opposite. I think the effects of lionfish are likely more concentrated on catch reefs. They're usually in higher densities on these reefs than on the continuous reefs. But one of my lab mates, Alex Davis, is actually looking at some of their effects on larger patch reefs and more continuous reefs, especially in terms of their movement. And it seems that some of these patterns are holding, correct me if I'm wrong, Alex, where there's hot spots of lionfish density, and then at dawn and dusk, they're kind of moving out around the reef and into the surrounding areas and then coming back. So, I think some of the results translate, but it would be great to replicate more of these studies on continuous reefs because there are a lot of different factors that go into that. Yeah.
Hey, Casey, great talk. Do you have any sense of how far they're going when they're going off at dawn and dusk? Like, obviously, it sounds like they're going down to 1,000 meters.
Yeah.
But is that something that's like a migratory thing, or?
Yeah, so I don't know exactly how far they're going. They've been shown through tagging studies to exhibit limited movement of several hundred meters apart, but that seems to be more when they're at one reef. Sometimes they can swim a few hundred meters and then they start living on the other reef, whereas these seem to be more them going out and coming back. All I could tell was that they had effects out to at least 30 meters from the reefs, but beyond that, I wasn't able to quantify their movement. So it would be really awesome if we could have an array out there and actually acoustically track their movements or something like that, but maybe the next PhD I'll do that.
Yeah, that's good. A follow-up to that was, are they putting anything on like bads or anything for you that you could sort of do an experiment like that?
Yes, I know there's one person, lad, who works for Reef, who's been experimenting with doing more tagging studies. So far, it's usually just been like manual streamer tags and not much else. Unfortunately, once you get on the really rugose reefs, all of the pingers and stuff don't do much good in those habitats. But to my knowledge, no one has done it too much yet. But yeah. That's a really good question. I don't know of any studies looking at that. So far, most of the studies on lionfish have basically showed that they eat a lot more and can eat a lot more, but I don't think anyone's transferred it down to the energetic side. Correct me if I'm wrong, lab mates. Yeah, but it would be really interesting to look into whether they have different requirements. Oh, yeah.
Maybe you said this, but why do they follow each other in a line out here?
I don't know. I mean, one thing is, in addition to the higher competition for food at higher densities, it could be that they're moving off the reef kind of in groups as some sort of latent fear of predation, because it's really risky to move away from the shelter of the reef. Now, lionfish don't really have to worry about predators, especially in the Atlantic with their venomous spines, and they're basically not eaten. But if they have some sort of innate fear of predators, maybe traveling in a group is safer than traveling alone. But yeah, it was really bizarre that they were doing that. Dave, then Kevin.
Nice job, Lisa. You showed that lionfish had an effect on recruitment. This might be out of the scope of your studies, but did you find any behavioral effects on spawning, site preference, or things like that for adults?
For the adults of other reef fishes or the adults
Correct,
of
other reef fishes.
Yeah. So I didn't look into that. If you come to Ty's defense in a couple weeks, she looked at the effects of lionfish on parrotfish grazing behavior, and Alex has looked at whether bicolored damselfish courtfish is affected, and I won't give away their dissertations, but there's some there's some mixed evidence. Most studies have found that prey fish are basically naive to lionfish and don't care at all whether or not they're there, with a few exceptions to that statement.
Although they care when they get eaten.
Well, presumably.
Let's thank Casey.
Oh, sorry.
And the committee will meet in Dornfeld across from the biology office.
Mark, can I say something quick?
Oh, Kevin.
You guys hang out for a second. I just wanted Casey to expound on something. She sold herself a little short in some of the information that she related. Give the crowd an example of the number of dives you would log in a typical summer.
Oh.
If you can sort of remember.
I don't know. 5 to 10 a day
Right,
for
so-
90 days.
I can help you out. These folks are logging typically in the range of 350 to sometimes close to 500 dives per summer. And you multiply that times the number of people in this lab and the numbers of their assistants and the numbers of years that they've been doing this work, and you end up with a pretty remarkable field record. So it's been a pleasure for me to work with these folks. This is sort of the end of an era when this Hickson lab kind of leaves and migrates out. So when you see these people, think about the thousands of hours they spent actually underwater, the hundreds and hundreds of times they handled these venomous fish. No incidences, no problems, no troubles. So it's been a pleasure working.
We can say that now.
This is Kevin Booth, the Oregon State University Diving Safety Officer.
I lost my hair before I came out. That was...
Thanks, Mark.